Hi everyone, this is the problem solving session 6 for basics of biology, and today we're going to study about immune system. So immune system is the line of defense for your body. It involves multiple kinds of cells and the components of your systems that are involved in defending the cells, defending the body against various kinds of pathogens. And the immunity can be divided into multiple types so that we can understand the functionality of the system as a whole. So the first kind of classification for immune system is the innate and the adaptive immunity. So innate is something, like the word itself says, that it's something to be born with. So innate immunity means the components of this system will be the molecules and the cells that you are already born with. So these, and that's why they are body's first line of defense. So you can see here that the innate immunity consists of many different kind of cells. For example, macrophages. So you have macrophages, then you have natural killer cells, then you have dendritic cells, neutrophils, basophils, and eosinophils. So all these are cells which are so let's say you are attacked by a pathogen, these are the first line of defense. So they will be their first response of the body to those antigens. And then you also have T cells and natural killer cells, which are also part of the adaptive immunity, which is the other kind of immunity. And innate immunity, as I said, they are the first to respond, so they have to be rapid in nature. So they are also called the rapid response. The second kind of immunity is the adaptive immunity. This immunity is developed when there is a, like, so they you acquire these components and molecules once you are affected by a pathogen. So they are acquired immunity and they happen after the birth, but they develop throughout life. So each time you encounter an antigen, each time you are falling sick, your adaptive immunity is also developing itself. It is there is something called memory cell. So those memory cell will keep in mind that, okay, this antigen has already affected me once, so the next time that antigen comes and binds to you or affects you, you have your memory cells, they can go and attack these pathogens. But they have developed, they are not you, they're not something that you were born with. So that's why they are slow response, because they will occur after a certain period of time once you are affected by the antigen. So that's why they are also called the slow response. So this was about the nature of the response, how they are when they are present in your body, and how they are acquired or developed during your lifetime. On the basis of that, the classification is innate versus adaptive. But there is also another class of immunity, or the classification of immunity, which is passive immunity and active immunity. So passive immunity means that the antibodies, so antibodies are the um, main component which binds to the antigen and leads to the killing of the antigen or the destruction of the antigens. So antibodies, if you are, so in this condition here where you see the B cell which produces the antibody, so antibody, if your body is synthesizing or producing on its own actively, that is called active immunity. But in passive immunity, the antibodies are pre-made. So you are administered passive immunity, or you're administered antibody which are already synthesized somewhere else, and they are being transferred or infused into your body. So that's why they are passive immunity. An example of this is during feeding, the child gets antibodies from their mother's breast milk. So that is passive immunity, because those antibodies are not synthesized by the child's body, but they're getting it from the mother. But when the child, after encounter with a pathogen, starts to synthesizing these antibodies in its own body, that time it is called active immunity. Okay, now that was adaptive and natural or innate immunity. Then we had passive and active immunity. The other kind is of humoral and cellular immunity, and these are your adaptive immunity. These are type of your active immunity. So like I said, that active immunity are, because they involve your adaptive immunity, they are slow. So they're slow response, they have lag period. but they are active against a wide variety of pathogens. And as I had mentioned, they involve immunological memory. So we'll see how that happen. But you have something called humoral and cellular. Cellular and mainly by the like cells, and humoral means via these flowing antibodies. So humoral basically is guided by the antibodies, while cellular are guided by different T cells. So if you just first look at humoral part, you will see that we have B cells. These B cells have BCRs, which are called B cell receptors, and the antibodies on their surface. So these antibodies will bind to their respective antigen, and once they bind, they start proliferating. So these B cells will start multiplying and give rise to two kind of cells. One is memory B cell and one is plasma cell. So the plasma cell will keep continuing synthesizing these antibodies, let's say in this case IgG, and so these are very specific to the epitope that was bound here, or the antigen that was bound here. So all these will bind to the same antigen, but this memory cell here will keep a memory of that receptor region from the antigen, and it will later start proliferating if there is a second encounter when it is not the plasma cell is not active anymore. So that's how you keep memory for the active immunity. Now, in cellular immunity, you have T cells as the major component, and these T cells are also of two types. They are called either CD8 plus T cells or CD4 plus T cells. 4 plus means which kind of CD they contain. So CD are basically cluster of differentiation. They are present on the membrane or surface of the T cells. So if they're this cluster of differentiation is of 8 groups, and it is CD8. Then these are T cells. They're also called cytotoxic cells. So CD8 cells become cytotoxic cells, while your CD4 cells are generally called the helper T cells. So they help in the B cell response. So what happens here is these T cells, they will have something called MHC class 1 as well. MHC means major histocompatibility complex. And what does that mean? That helps them to bind to several antigens and other cells which are responsible for immune response. So here, this is dendritic cells, and CD8 has MHC class 1. You can see here, and that helps them to bind to the dendritic cells. And then this dendritic cells, so they can then so if this they're bound to dendritic cell by this, the dendritic cell can also bind to the CD4 T cells via the MHC class 2, and the MHC class 2 TCR cells, once they have bound to antigen, they will on their other side they will have these what do you call CD40 and TCRs that will help them to bind to B cells. So they are also, once they have bound themselves with the antigen here, they proliferate the B cells to give rise to more antibody. And on the other side, CD8 plus themselves can become cytotoxic T cells to kill the infected cells. If not antibody, if not kill infected cells, then is this interaction between the dendritic cells and the helper cells can give rise to inflammatory responses by the synthesis of or the production of interferons and interleukins, all the components that are responsible for inflammation responses. That's how they work together. So if I have to take you step by step in each of these immune systems, so first is humoral or the antibody mediated immune system. So we have the extracellular antigens here. And then we have a B cell which binds to this antigen. And this has to be very specific because B cell has something called antibody, and antibody and antigen interaction has to be very, very specific. And then there is a T-dependent B cell. So a T-independent B cell requires cooperation from TH cells. That just we have seen that how a helper T cell can help proliferation of the B cell. And then the B cell proliferates, and some of them become plasma cell and some of them become memory cell. Then the P cell, often with stimulation from the helper T cell, they will differentiate into plasma cell that will generate the antibodies, and then would be memory cell. And this memory cell will be responsible for a response when there is a second encounter with the same antigen. And for cell immunity or cell mediated immunity, you have the exposure to antigen, or you can be having intracellular antigens which are present on the surface of a cell which is infected by a virus or any other pathogen. There, a T cell will bind to the MHC antigen complex on the surface of the infected cell, and then it will activate the T cell response. So this is a cytokine response, are all those interleukins and interferons that we had seen. And it will also activate macrophage for enhanced phagocytic activity, so that these infected cells are ingested and they are lysed there. And CD8, they will become cytotoxic T lymphocyte and they can induce apoptosis. So you can see how these cells are killed. They're killed either by a phagocytic effect or by cytotoxic effect or by B cell mediated interactions. So we'll first talk about antibody in detail. So antibody is something like this. It has a Y-shaped structure. It has two chains. So it has one heavy chain and one light chain. So the heavy chain is shown by the green color here or blue color here, and a light chain is shown by the light color or pink color here. Now, to understand the structure here, you see that there are two light chains and two heavy chains for each antibody. In the light chains, you have two parts. The N-terminal region has the VL. So V means variable. So this is a variable portion of the light chain, and this is the conserved portion of the light chain. Similarly, here you have the variable portion of the light chain and the conserved portion of the light chain. And here, this is the heavy chain. So in heavy chain, you will have one portion which is variable. But then there are three domains which are conserved, which are called CH1, CH2, and CH3. So these are conserved domains. Now, these both domains or these both strands are connected to each other by disulfide linkage. And even the light and heavy chains are also attached to each other by these disulfide bonds and non-covalent bonds. Now, the most important thing is why they are variable and why they are conserved. So these variable regions are the ones that bind to the antigen. So the region by which the antigen binds to the antibody is called the epitope. So let's consider here this region. This is an antigen, and this region is where it binds to the antibody. So this region is called the epitope, and respectively the region which on the antibody binds to the antigen is called the paratope. So if it is the region that has to be bound to the antigen, so this region will be known as the paratope. So the antigen antibody introduction is actually mediated by the epitope and the paratope. Epitope of the antigen and paratope of the antibody. Now, why is this V and why is this H, or why is this variable and why is this conserved? So variable means the structure or the sequence of these domains are always changing. And can you guess why that would be important for an antibody? Why doesn't it why does it want to always change its binding site? So the easy answer or easy way to think is there are so many antigens in the nature that can come and bind or infect you. The antibody has to bind to all those antigen, and how does it do it? If it can bind, if and it has to be very specific, the binding has to be very specific. That means every time a new antigen comes in, the antibody has to bind it perfectly. So that's why these regions are variable. They keep changing every time so that they can go and bind to the antigen, and they're not conserved, because if they're conserved, their binding affinity or their binding to a particular antigen is restricted because of their limitation of the sequence. So that's why these are variable region, and they give rise to humongous kind of combination which can bind to any different antigen. But the other region, which are these regions, these are conserved because that's what gives them the structural stability. These conserved domains have region that can bind to each other and give rise to this wide kind of structure. And also another important factor is, so this in the antibody, this region which consists of the variable portion of the heavy and the light chain, these are also called the Fab domain, which is fragment antigen binding. So because they bind to the antigen, so that's the antigen binding fragment, so you call them as a Fab domain. And these are the crystallizing domain because they are the heavy part of the antibody, so they are crystallizing domain, and that's why they are called the FC domain. And once you bind, your antigen has bound here, antibody has to bring out effects like probably take it targeted for phagocytosis, probably targeted for killer cells. So those interaction with other cells happen because of the conserved region, and that's why they are also called the effector region, because they bind to the effector cells. Now, you generally, the IgG are of mainly five types: GAMED, and these individual antibodies have their own activity. For example, IgE is for your generally for the allergic responses. Then you have one of the antibodies which are IgA, which are transferred from the mother to the child during breastfeeding. So all these antibody have their own sizes, have their own functions, have their own different subunits as well. But and they serve different purposes. So some of them will be involved in the initial responses, some of them will be involved in the secondary responses, some of them will be only restricted to histamine or allergy reactions. So that's how the diversity or repertoire of antibody is present in your body. Now, as I had mentioned, and we have already seen that antibody has to be produced by B cell in response to the pathogen which is infecting. So there is always a lag period during the antibodies production, because in the initial phases the other cells are binding to the antigen, and it's only when they come and bind to the B cell and the B cell gets activated is when the synthesis reaches an exponential phase, and then after a certain point of time it reaches plateau and then it starts declining. So that's the overall procedure by which the antibody is produced. And so we'll go over that step by step. So here, like I've mentioned, that whenever an organism's immune system encounters a foreign particle for the first time, macrophages are the first one to go and interfere. They capture to break them down, and then they pass it on to B cells. So all this is happening while the lag period is going on. Now, once the antigens are presented to the B cells will start the production of new antibody by the epitope and paratope interaction. So these antibodies that will be synthesized by the B cell will now all of them have unique paratope. Then each lymphocyte of B cell, so B cell is our kind of lymphocytic cell. So the lymphocyte, each cell which is now generating an antibody against an unique epitope. Then once this encoding and when they have started just synthesizing the full antibody, these antibodies are released into the bloodstream, and which go and bind to the respective antigen wherever they encounter them. And what happens after they bind them? They eliminate the antigen from the body. And how do they do it? Like I had mentioned, that they can either do it by direct attack of the antibody on the pathogen, or by binding to the pathogen surface and then sending out signal to the other components of immune system to come and eliminate. So either it can itself start inducing the lysis of the cell, or it can call upon macrophages or lysosomes or any other procedure which can lead to lysis of the cell. And as I mentioned, they remain in the body as memory cells. So the next time when these antigens encounter, these are the first cells to respond to them. So yeah, that's what we have been telling. Now here, one word that you can see is isotype switching. This is where the diversity will happen when they keep switching their all the domains, and then they can generate diversity to bind to the antigen. Okay. So now antibody, because they bind to antigen, they are the type of how many antigen they are binding and how the type of antigen they're binding. On the basis of that they are classified into monoclonal and polyclonal antibody. So poly and mono, as the name says, mono means one, poly means several, and clonal means the type. So to understand first very briefly, if you have a polyclonal antibody, it can bind to different antigens on its surfaces, because it will have the paratope region for all those different epitopes, but the individual interaction will be very specific. But monoclonal antibodies means they are all clonally amplified antibodies which will all bind to a single epitope only. So you can see by the image here that let's say this is an antigen and it has several epitopes on its side. So if antibodies are polyclonal, they can bind to different regions based on their different paratopes. But if they all of them bind to a single domain or single epitope, that is called the monoclonal antibody. And brief differentiation between them is, so how are they produced? So in generally when you have an antibody synthesis, they are all invariable because their different antibodies are binding to different epitopic region. So you have polyclonal antibody synthesis. For monoclonal, it's generally in vitro. You deliberately are able to produce antibodies which are monoclonal, which are very specific to a particular epitope. So the next is target. So as I mentioned, monoclonal target one epitope and polyclonal targets multiple epitopes. Because we are synthesizing it here, uh, so the monoclonal antibody is expensive and polyclonal is relatively cheap because you have to be very particular in the production of monoclonal antibody. So that's very expensive, and because it involves a very highly complex technology as compared to the polyclonal, which is, if you want to produce polyclonal antibody, the vaccination process is very easy because you just need an antigen to be you to administer into the like the individual. And batch to batch, so let's say you have produced antibody at different time or at different batches. For monoclonal antibody, because you're targeting one single region, the variability is very less. All the antibodies are identical, while in the polyclonal there is large variability because there are multiple epitope sites involved. You have this large variability in the antibodies. And monoclonal can be very large because we'll see how does that happen, while polyclonal, because it's involved, it's limited. And cross reactivity, again, polyclonal is possible because it has epitope, so you can use it for neutralizing some other antigen which is not for the one against which it has been raised for. But for monoclonal, it is very low because of the specificity of the epitope. So first we'll see polyclonal antibody. So as I mentioned that you have this, in this you are doing basically immunization and synthesizing it. So generally this is rabbit or horses where you inject the antigen into the organism or the animal, and then the animal synthesizes B cells because the antigen activates the B cells and they proliferate, they produce the polyclonal antibodies and the plasma cells, and then these animals are bled. So from the serum you basically harvest all the polyclonal antibody and then they use it for the downstream purposes. Now there are two important words, antigen and immunogen. What you injected here has to be an immunogen, but what we were talking about all this while was something called antigen. So there is a very basic difference between antigen and immunogen. So if you look at the word itself, immuno means immune response, something to do with the immune response. Antigen means something which is against the, like, which is not from the body, which is something from outside. So if we differentiate them, antigen is a substance specifically binding to the antibody or a cell, B cell or T cells. Immunogen is an antigen, however, which is capable of inducing an immune response. So you would think that antigen is something which is outside cells. So anything that inside enters the body from outside should be able to give rise to immune response. That's why all the antigens should be immunogenic. But you see the second point, it says that antigens could be immunogenic or could be non-immunogenic. But immunogens, if you call something as immunogen, they are always immunogenic. Now, this is a very important line. Not all antigens are immunogens, but all immunogens are antigens. Now, why does that happen? That happens because antigens can be proteins, polysaccharide, lipids, or nucleic acids, while immunogens become morely like normally they are proteins and large polysaccharide. So, because of the size difference or the compositional difference, antigens could be very small. for the body to detect itself as something which is like to detect first of all, and then the body will elicit response against it. So for detection you have to have a proper cutoff for size. These antigens could be very small, but when you, so when does an antigen becomes immunogen? When it is able to be recognized by the body, that there is a detection that there is something from outside into the cell. And what happens for them is that you have something called haptens. So these haptens, they are low molecular weight compounds which bind to antibodies, and they bind to these antibodies, or separately you can also put them with antigens, so that their molecular weight increases and they are now immunogenic. So if you want to put something here as an immunogen, so that because you are introducing something not at a natural condition, you need to put them with haptane so that they become large molecular weight, and so they can generate this immunogenic response that you want in the organism. So that's why you have to be sure that whatever you are injecting is actually an immunogen and it is not just an antigen. So generally what you do is you amalgam, or you put these haptens with these antigen, and then on top of that you add some adjuvants. So you what does that mean is they augment the effect of vaccine by stimulating the immune system. So you make sure that whatever antigen you have introduced into the organism is able to elicit the response, and the response is also at a higher level. So you give haptens and adjuvants along with your antigens. So that was about polyclonal antibody. Now about monoclonal antibody. So you can see that monoclonal antibody requires lot of technology. For example, here you will have fusion of two different cells, then you have selection medium, and then you will have in vitro culture. So you see the number of steps for the synthesizing monoclonal antibody has gone up, and that's how your complexity has been introduced, and that's why these are complex, like these, because of the complex steps involved here, they're very expensive. So we'll do this in detail here. So monoclonal antibody is produced by something called hybridoma technology. Hybridoma, as the name says, there has to be some hybrid of two cells while you are synthesizing the antibody. And what are the hybrids of? Those hybrids are of B lymphocytes and myeloma cells. Now, individually, what is B cell? B cell is the cells which synthesizes antibody. And what is myeloma cell? Myeloma cells are those cells which are capable of infinite growth because they are cancerous cells. They're oma. And the myeloma cells that are used in this technology are generally bone marrow tumor cells. So they are tumor cells, so their property is to grow at a very faster rate and grow infinitely. While the B lymphocyte that have been harvested from the animal that was already introduced by the antigen that we need. So B lymphocyte will be able to produce antibody specific to that antigen with the required epitope. Okay. So you have these two products and then you fuse them. And you fuse them such that the product that has become now you have a B cell, you have a myeloma cell. If you fuse them, what becomes is a antibody producing cell that can grow infinitely. So that's the idea here. So that's what it says, that the fused cell has the ability to produce large amount of pure antibody and as well as grow or multiply indefinitely. Now, just to go over the steps here. So individually, first is myeloma cells. You've taken the myeloma cells, you've incubated them, and you have made them HAT sensitive. What is HAT will come in the next slide. But remember they are HAT sensitive. These myeloma cells are HAT sensitive. While for the B lymphocyte, you've immunized a lab animal and you have given booster doses to augment the immune response, and then you isolate the B lymphocyte. Then you fuse these two cells in PEG media. So you PEG is polyethylene glycol. So you fuse them. Now what will happen? You will have the cells are supposed to fuse with each other. That's what there's no irrationality. So a B cell can fuse with a B cell, a myeloma cell can fuse with myeloma cells. Some cells might not even fuse. But then there are some cells that it's exactly what we want. We want the B cells and the myeloma cells to fuse, that will give rise to hybridoma cells. You grow these in HAT media, screen the cells which are the true hybridoma nature, and then once you know that these are hybridoma cells, you culture them either in vitro on the cells, or you put them back into the animals so that you can upscale the production. So we'll now discuss about this important step where we are able to differentiate between the different kind of fused cells. We need the true hybridoma cells and we don't need any other fused cells. And how do we do that? So that's where the HAT comes into play. HAT means hypoxanthine aminopterin thymidine medium. So these cells are grown in HAT medium, and that serves as a screening background for identifying the hybridoma cells. Now, first, we'll try to understand what is the principle here. So, in any nucleotide synthesis, so for survival of cell, they have to produce nucleotide because we know that they have to replicate. So nucleotide synthesis starts from sugar. So you have a simple sugar, they are taken by multiple pathways to give rise to nucleotide. Now, if in your media you have given aminopterin, this procedure where they can use simple sugar to give, because you will give sugar in your cells for in your media for sure. But if you give aminopterin, this de novo pathway of using the sugar to produce nucleotide is inhibited. So any cell which is grown in aminopterin media and with sugar will not be able to produce the nucleotides. First, we'll know this much. Next, you have also added hypoxanthine in your medium. But there is a, so if not sugar, the organisms can, or the cells can use this hypoxanthine to give rise to guanine. Guanine is again a nucleotide, and this is by the HGPRT enzyme. So if not de novo pathway, you have given in the media hypoxanthine, so they can use salvage pathway to give rise to the nucleotides that they require for survival. This is the idea. Now here you have B cells and you have myeloma cells. You have made myeloma cells as HAT sensitive, means you have, this was anyway working to not working so that you cannot produce any nucleotide. You have made the myeloma cells on top of that HGPRT negative. So you have removed this enzyme from myeloma cells. Now because there is no HGPRT, these myeloma cells will not be able to use hypoxanthine from the media to give rise to synthesize guanine. Now you imagine myeloma cells, you have put that on HAT media. Because of aminopterin, it can't synthesize nucleotide by de novo pathway, so it is bound to die. It also doesn't have HGPRT, so it can't even use salvage pathway, so it is bound, it is doomed to die in this media for sure. Now B cells on the other hand, they are normal cells, they will have their HGPRT. So even because of aminopterin, they can't use de novo pathway, but because they are normal and they have the HGPRT, they will be able to use hypoxanthine to synthesize guanine and survive. But B cells are not cancerous in nature, they have a certain lifespan. So after a certain point of time, they will also die. So myeloma cells will die, B cells will also die. But now you have hybrid cells. Now hybrid cells have combined, are fused from B cells and myeloma cells. B cells have HGPRT. So these hybrid cells have HGPRT, and with their HGPRT, they can synthesize nucleotide. And because they are myeloma cells, they are cancerous cells, they can grow indefinitely. With this synthesis, they can continue to grow in the media. So the HAT media, you will not have so if you see here this flowchart now, we have B cells which are HGRT positive, and myeloma cells which are HGPRT negative. Fuse them up, you get only B cells. Only B cells will grow on this media of HAT because they have HGPRT. They will be able to use hypoxanthine. But after a certain point of time, they will die because because of their normal lifespan. Myeloma cells, on the other hand, because they are HGPRT, they could have grown indefinitely had they had this enzyme. But because they don't have this enzyme, they will not be able to follow the salvage pathway, because of which they will die no matter how cancerous they are. But then you have this third category of cells, which are the hybrid cells, which can grow indefinitely because they are myeloma cells, and they can grow in this cell because of the presence of HGPRT. And that's how you will only have hybrid cells growing on your HAT media. Then you can use these cells and harvest them, and then you can upscale the production of these cells to get the monoclonal antibody. So that's how monoclonal antibodies are synthesized or produced at the lab scale. Now, so this was all about how we produce the antibody. We'll now talk about how the antigen and antibody interaction takes place. So as I've already mentioned that antigen and antibody interaction are very, very specific because of this sequence specificity here. This epitope and this paratope not only sequence, they will also have a proper log jog conditions wherein they will have structural affinity as well. So they can bind to this antigen with much more specificity. So this gives rise to the antigen antibody complex. Now, antigen antibody complex can be or the reactions can be looked or can be identified by different reactions methods, where you can see if their antigen antibody complex has formed or not. And for that you have precipitation reaction, agglutination reaction, complement fixation, immunofluorescence, or ELISA. So we'll go over these one by one. The first is precipitation reaction. Very easy to understand that if you have a, like in a solution, if you have antigen antibody complex formation and that becomes a precipitate, that gives you an idea that there is presence between antigen and or there is a presence of antibody for that antigen. Now where do you use these kind of reactions? These are generally tests which are used in medical setup to understand if you have any antibody in your system for that particular antigen, or if you have that antigen in your system, so that you have like what is the infection cause probably. So you use precipitation reaction where you rely on the formation of an insoluble precipitate which is formed because of the antigen antibody complex. And this procedure can be carried out in liquid form or in gel. So if it's in liquid, it's easy to understand that you will have a increasing amount. So you in a tube you have a constant amount of antibody, but you increase the amount of antigen as and when the antigen antibody reach equilibrium concentration, they form bonds and they precipitate. That gives you an idea, okay, this many antigen was present in the in the sample. Then you also have gel precipitation where you have these agar gels or any other gels. In the gels you have this antigen antibody, they are, so they're diffusing, and then when their zone of equivalency, when their concentration are almost increased, you can see a visible precipitate forming on the gel layer. So you visually see the precipitation in the gel's continuous gel surface. So that also gives you an idea that if there is presence of antigen or antibody. Next is something called agglutination reaction. So instead of precipitating, you have these little clumps that are formed where you can see that, okay, antigen antibody interaction has taken place. And these clumps of could be of cellular antigen, which so if you add antibody, you can see the clump formation. And this kind of reaction is called agglutination. So in this condition, you call them agglutinins, the antibodies which are actually carrying out or bringing forward the agglutination reaction. Here also you have two kind of tests. You could do it on slides or you can do it on tubes. So you can see here, so this kind of test is called Vidal test, where on these kind of slides you will have the antigens and you add antibody on top of it, and you see this kind of clumping forming. So if you see clumping, so you can see in the negative, because of no clumping, you see all of them being spread around in the slide. But here in the solution you can see this clumping formation, so you can say that this is positive and this is negative. You can do the same kind of reaction on the tubes, where again the same principle will happen. So you can for this you can see that you have antigen suspension, for let's say for example, and then you use the antibodies in a serially diluted form to see exactly where the clumping starts happening, to know exactly what concentration of antibody is present, and that is called the titer value, the value which is actually able to the concentration that is able to bind to the antigen. And this kind of test of agglutination is generally used for blood group typing. So blood group, as you know, you have drop of blood, and blood have different antigen that gives rise to antibody. So let me write it here. So if you have A antigen, that is A blood group. If you have B antigen, that is B blood group. Where are these antigen? They are present on the cell surface of the RBCs. If you have both A and B antigen on your surface, then you have AB blood group. And if you have neither of these, then you have O blood group. So the kind of antigen that is present on your surface gives rise to the blood group. Now, for these tests, what do you generally do? And you also would be calling something as B plus and A plus. So what are those plus? Those plus are something called RH factor. So if you have RH factor, if it is present, then your blood group is A plus. If your RH factor is absent but you have A antigen, then it is your A minus. Similarly, B plus and B minus, AB plus, AB minus, O plus, and O minus. That's the different kind of anti blood group that we have. So for testing the blood group, we generally what is done is agglutination reaction. So what happens in this is, so you have these droplets, and on top of this you add these antibodies which are anti-A. So antibody for A antigen, anti-B, anti-D. So the RH factors are also called D. So you have the anti-D, and then you have control where you don't have any antibody. Now here, so what I'll do is I'll first go over this profile and then we try to see if we match it. So you have no, so if your agglutination reaction has to happen, the antigen has to be present. So you see in which of these drops or droplets you see the agglutination reaction happening. So in this, there is no agglutination for A, no for B. That means there is no A or B present in the blood, but you have agglutination for your anti-D, and control doesn't show any agglutination, which is correct. This is a control which says that the reaction, the experiment that we did is correct. So because there is only one well which shows agglutination, that means it is positive, RH positive, but the blood group is O. So that says it's O positive. It had to be, if it had to be O negative, this thing should not have shown agglutination because you don't have the RH factor, and that's what happens in O negative. Now in A positive and A negative, you will have the anti-A while showing the agglutination. So you show that, and if you have anti-D also showing agglutination, you are A positive. If not, you are A negative. In B positive, only the B well should show us agglutination, and anti-D should give us if they are B positive or B negative. If both the vials of A and B show agglutination, that is AB. Again, if the anti-D shows, so basically all the three vials show your agglutination, they are AB positive. But if they are one that shows that which does not show agglutination, then it is AB negative. But you have if agglutination even in the control, if you see agglutination in any of these vials here in the control column, then you know that your test has gone wrong somewhere. So that test is invalidated. That's how you do blood group typing in medical setup. So agglutination can also be done by something called Coombs test, in direct and indirect way. So in direct, you have these antigens directly, which are coated with the antibody. So you want to check if these antibodies are present. So you put in the antibody. So let's say here the antibodies are IgG. So first you harvest these antigens which are with the antibodies, and then you give the anti-IgG to form these agglutination, and you can see that okay, if you have added anti-IgG and you see agglutination, then you know that there is anti-IgG coated RBC in sample. But for the indirect one, what you do instead is, so you don't use the antigen like the RBC which is coated with the antibody. You want to rather see the antibody in the serum. So you have the reagent. So you have the erythrocyte. You take the erythrocyte, and then you use the anti IgG. So here, So you check for unattached antibody. So in this test, first you take the patient serum, which is the normal red blood cells, and anti-serum to human immunoglobulin is added here. And if you see any kind of agglutination, then you know that there is an antibody flowing, freely flowing in the patient serum. So that was about agglutination. The other kind is complement fixation. So if there is an antigen antibody complex, and that can they can also bind to complement. So you can detect if there is an antigen antibody complex by the addition of complement proteins. Complements are additional protein which help in the immune system. So to check if antigen antibody is present by this method, what you do is you have antigen, you put it in a solution which contains the antibody, then there is you allow the complex to form. If your sample has antibody, so this is a sample that we are trying to test if it has antibody or not. So you add your just a second. Yeah. So what you do is you want to check if in your sample you have antibody. So you take the sample considering that they have antibody. Now you add antigen, you allow the antigen antibody complex to form. Then you additionally add complement. So this complement will go and bind to the antibody antigen complex. Then to check if your complement is free or not, you add RBCs, and you add RBC on top of it, and then you also add your own version of antibody. Like if you're just sheep RBC, you add an antibody to sheep RBC. That in complex goes, and you have because this is your RBC, if your complement binds to this, it will initiate the hemolysis. So you will see hemolysis happening here if the complement is free. But because in this system it has already bound to the antigen antibody complex, it will not be able to bind to the second antigen antibody complex that you have added, which is called generally the indicator system. And because it doesn't bind to the indicator system, there is no hemolysis that happens. You see that this is a positive result. But if in this case, for example, there was no antibody for it to bind to in the earlier case, so when you add this second indicator system, or when you add the indicator system, the complement goes and binds to that antigen antibody complex and give rise to hemolysis. That's how you can tell that in your sample was there antibody present or not. The next is ELISA, and we have done ELISA many times, but once another similar concept is RIA, which is called radio immunoassay. This is based on this very similar idea. So in RIA, you have the antibody and an antigen. Instead of being tagged by an enzyme, it is tagged by a radio labeled element. So you have radiolabeled antigen, and then you have the normal unlabeled antigen. So what you try to see is when you add the sample, how much displacement of this radioactive antigen has happened due to the presence of this unlabeled antigen. And based on because these are competitively displaced, the radiolabeled antigens go into the supernatant, and then you check the radioactivity before the binding and after the binding, and try to estimate what level of antigen is present in the patient sample. So, but a modification to this, instead of using radio labeled antigen, you rather use an enzyme or a secondary antibody which is tagged with an enzyme to check for the interaction between the antigen and antibody. And we have done this in the previous sessions. So that was all about how you like how you detect the antigen antibody complex, especially in a medical setup. Now, this is what happens after an antigen antibody complex is formed, and how the infected cells are removed from the system. So this is the step is called phagocytosis, and you can see here in the animation how does it actually take place. So I'll just mention it from the figure. You can simultaneously see the animation as well. So you have these are the infected cells. These are the cells which are infecting the organisms of the pathogen. So you have the receptors which engulf them, from a vacuole against like from a vacuole against these surface, and they are taken inside as a phagosome. This phagosome will lies with lysosome. Lysosome has many enzymes that can kill off these they can lyse these bacterial cells. So once the phagolysosomes are produced, the enzymes are secreted from the lysosome that can disrupt these bacterial cells, and once they are killed, they are then released by exocytosis as soluble debris that are then removed from the body. That's what happens once that's the clearance process of these infected cells from the body. And these cells which perform phagocytosis are generally neutrophils and macrophages. So to explain in details, so this recognition that okay, this cell has to be tagged for phagocytosis is generally aided by something called opsonins. So if you have opsonins, then the or the host plasma protein, then you mark the bacterial surface for this phagocytosis where the neutrophils or macrophages can recognize them. And these opsonins, which are, like, they are targeting or they are marking the cells for phagocytosis, they are generally immunoglobulins or the complement proteins. So if you see here, what happens is you have these antigens, then the antibodies, and then you have these antibodies, as we have said that they are opsonins, so they can be taken inside and then they are glyced. To understand a bit more detail, you have these complement protein here like C5 or C3b, and they are responsible for this attachment to the phagocytosis cell. For example, if it is a macrophage or natural killer cell, neutrophils. So you have these antigen antibody complex already formed. The C3b is the complement protein which is on the antigen. They will be responsible for the recognition and attachment, and then these pseudopodias will come because of actin, because actin helps in the elongation. So they will be elongated, they will form a circle around it, they will be engulfed, and then the phagosomes will lie with the lysosome, and then will be digestive enzyme that will lead to the destruction of these cells. So that's the overall idea of phagocytosis. And to understand in more details, once they are internalized, the phagosomes will, once they are formed being the phagolysosome, there is a decrease in pH. So in a decrease in pH, the condition become acidic. Many of the enzymes which are have hydrolytic nature, they get activated, and then they will lyse these cells. That's how you the main action is because of the change in the pH. And now you can also check for phagocytosis via various assays. For example, this here. So what you generally do is you use fluorescence to check if there is phagocytosis happening inside the system. Now you have green fluorescence and red fluorescence to demarcate between the two. How do you do that? So your green fluorescence is generally for live cells. Any cell which is alive is able to take up that protein, which is fluorescent, which is green fluorescent protein, and it will fluorescent green in color. And then you have also put red fluorescence on your your substrate, the antigen which you want to be seen if it is if it is taken up by phagocytosis. And if you see something like this, green with the red, then you know that the live cells are taking up the substrate. That's why you see more of red fluorescence in the green zone. But and how do you check for control? How do you know that this is not happening only as a problematic experiment? So I've told you about actin. This actin is responsible for this engulfment, right? So if in your setup you use cytocalysin D, cytocalysin D inhibits this actin activity. So what happens if there is no actin activity? These pseudopodias are not able to engulf. So this red fluorescence body does not actually go inside the cell, right? Because there is not any phagocytosis that is taking place. So you use cytocalysin D, and you should see less of red color and more of G color, because G means green cells, and the red cells that you see should be outside of these green cells, right? And that's exactly what you do. This is a control. You see that when you add cytocalysin D, The red cells are all outside, the antigens are not taken up by phagocytosis in comparison to the control where you see lots of red color with the green cells. So that's how you can, like by use of the fluorescence, you can see, check for phagocytosis in the cells. The other method that generally is used is called flow cytometry, which is also used for identifying different kind of cells undergoing phagocytosis, or just for the identification of what kind of cells are there present in the system. So flow cytometry are high speed, and they are able to scrutinize even at single cell level. So you have this big setup here where with which you can identify what kind of cell just entered, or what kind of cell are present in the system by just passing one cell at a time and recognizing that cell. So, and it can also measure the cells in solution, at least how many cells have passed per second, and it can do rapid analysis, at least characterizing millions of cell. And but if you do it at a faster pace, you don't get to know what are the morphological characteristic of the cell, and like where is it actually present. But you can at least get to know what is the amount of that cell present in the system. And it is again based on fluorescence intensity. So you have these fluorescent labeled antibodies which are detecting the proteins or whichever ligands you want to test. You rely on the use of fluorescence. So we'll go over this in a detail. So like flow cytometry is divided into three parts, or consists of mainly three components. First is fluidics, second is optics, and third is electronics. Fluidics is where your cell is actually going to pass, or sample is going to pass through this setup. Optics is where you actually have those emissions, the light emissions that we were talking about, the fluorescence. And then you have electronics, which will detect these emissions and let you know what was the profile of the cell that has just passed. So in fluidics, to go over in detail, you have these samples, transporting the sample to this tube, and once you this cell there these are sample is past this flow cell. These can either be, so here, while it has been taken to this path, after this you can sort also. So let's say you have to sort your cells according to the presence of the cell. So let's say you want to sort B cells from the other cell. So it can also be assisted with cell sorter, where you can put in the required filter, and you can say that if you see a B cell profile, you deflect the cell to the right and you put it onto this beam. Otherwise, you can just help all these cells washed away and you don't need that anymore. The second part is optics. So here you have the excitation lights, the lenses, and the filters that can collect the light that has come from the system and then pass it on to detection system, which is the electronic, which will give you the digitized signal. So to check the fluidics in detail, so you have the single cell suspension from a cell culture. So you incubate the cells in tubes or microtiter plates with unlabeled or fluorochrome labeled antibodies, and analyze here on the flow cytometer. So you have the cell suspension when they run through the cytometer. So there is a fluid here. This is called a sheath fluid, which so it will give enough of the force so that it streamlines the flow of cell as one per unit. So there are so many cells, but because of the pressure, because of the presence of sheath fluid here, the pressure only allows one cell to pass at a time from the nozzle. And then there is this tiny stream of fluid which takes this. So here, after this certain point of time, they will encounter the laser light. Now, when there is a laser light passing through it, the cell will get illuminated. And because of its feature, because you are just looking at single cell right now, you don't want to count number of cells, but you're looking at single cells individually. Then because of its properties, if its size and its granularity, how many granules, how granular it is, what kind of clumps it has on the surface, it will scatter the light. And the scattering happens in two parts. There is something called forward scatter and there is something called side scatter. And on the basis of this scattering of light, the light is then detected by the detectors, and on the basis of that, profile is generated. So So here the light is scattered from these cells in the forward direction, forward scatter, and that forward scatter of light would be due to the size. And if the side scattering is because of the granularity. So if you have granularities more in the cell, there will be more of side scattering of the light. But if the size of the cell is higher, then the forward scattering will also be higher. Now, because you have these two profiles, you can distinguish between the cell population based on the difference of size and the granularity, right? And then the light from the laser, as so here, if you have fluorophores, you will also have a fluorescence emission. Instead of this kind of peaks, you will have fluorescence emission to identify what kind of cells just pass through, because you have tagged the cells with certain kind of fluorophores. Now here on this scatter plot here, if you see, let's try to identify what kind of cells are present in the system. Now these cells here, they have very high forward scatter. So they're large forward scatter and very high side scatter. Means they are large cells, they are also granule, they are high granularity. So they are generally the granulocytes. Then you have monocytes. Monocytes have very high size, so their forward scatter is high, very large, but their side scatter is very low because they are less granular. Now compare this to lymphocytes, which are very small cells, so they have very little forward scatter and side scatter. So based on this kind of profile, you can see what kind of cells are present in the system or in the sample. Now once your optics has happened, the light emission has happened, the next part is these electronics, where you have these different photo multiplying tubes. So what they do is the light and the fluorescence that has come from these cells, they are split into defined wavelength, and then they are channeled by the filters and all these photomultiplier tubes at a specific wavelength that is detected finally and given out as an output. So as the fluorescing cell passes through the laser beam, it creates a peak, a pulse of photon emission, because that's how it is detected. And then how much of this emission has happened gives us an idea of how much of these cells populations are present. And what you do is then you see the voltage pulse area and you corroborate it with the intensity of the fluorescence that you have checked. And if you see a shift in the intensity of the fluorescence, then you know that there is a positive cell, and the area under the curve will give you how many cells are present in the sample. And this, checking particular cell for a particular sample is called gating. So gating means, let's say I have marked that I will only check for B cells, which I know how I have marked with certain fluorescence. So if I only look at the wavelength of a particular fluorophore, then I am gating the system for a particular cell type. So that's what you do generally in your analysis. So if you have a, let's say, phagocytosis, and you know that there is fluorescence for GFP and fluorescence for cytokeratin, you gate them and then you get the fluorescence profile. You can also try to look at how many cells of the cell cycles are in which states. So for that, you generally use propadium iodide for the cells as the marker, and then you try to see how many of G1 G2 S cells are present. You can also do this for apoptosis, where you generally have these FITC. So that's the fluorophore that you use, and you bind this to the one of the, so in apoptosis you will have cells with connect genes or any other marker, and you can tag those marker and see if the apoptosis is happening or not based on the granularity and the forward and the side scatter. So that's how you use flow cytometry in the detection of phagocytosis or any of the cell counting or cell identification mechanism. Now, we spoke about the phagocytosis, where you have these transport of the antigen into the system and then you tag them for lysis. We also had discussed about vesicular trafficking in the previous slides, and we had spoken about three organelles that are responsible for trafficking of any of these components inside the cell. And those are endoplasmic reticulum, Golgi apparatus, and lysosome. Lysosome, we have just discussed that they are responsible for the killing of cells. Golgi apparatus and endoplasmic reticulum are responsible for transport of cell from the cytoplasmic phase to the nuclear phase and from the nuclear phase to the cytoplasmic phase. So the endoplasmic reticulum being closer to the nuclear transport the material from the nuclear end to the cytoplasmic end, and Golgi is just after endoplasmic reticulum, so it can take in the right direction towards cytoplasm, or it can also transport from the cytoplasm towards the nuclear part. So here you can see that this is how in vesicles the transport is taking place. So if you see here that from the Golgi apparatus you have these transport vesicles, which are, so there could be, we have already discussed about it, there could be some proteins which are destined to be in the plasma membrane, and then there are some particles which are required to be secreted. So you have secreted vesicle that will go and be exocytosed from here, they will be regulated secretion. But then there you have some which do not require to be secreted, so they will go and be embedded in the membrane here. And so in vesicles only, either you have movement from the nuclear to the cytoplasm and then to the plasma membrane, or you can also have endocytosis and then you can have the same movement towards the inside of the cell. So for example here, this is the plasma membrane and this is the endoplasmic reticulum. Here is a nuclear and then the Golgi in between. So you have a plasma membrane getting some pinching off as an endosome, and this endosome is taken into recycled as early and late cycle, and then you have the endosome which is bringing the particles to you. Now from the endoplasmic reticulum, the same vesicles, they are transported to Golgi, and the Golgi and then give it to the endosome that can take it to plasma membrane. But it can also send the like the materials back to the endoplasmic reticulum. How does it do it? How do they ensure that the directionality of the movement is because of the presence of the markers on the vesicles? So, for example, if you have clathrin is mostly responsible for this recycling of the endosome and lysosome. COPE1 is responsible for getting the Golgi, the vesicles from the Golgi to the endoplasmic reticulum, while COPE2 is responsible for getting the vesicles from the endoplasmic reticulum to the Golgi apparatus. So that's how you determine the direction of the movement in such trafficking system. Now, this also we had already discussed that endoplasmic reticulum, because it is conjoined with the nuclear membrane, they are responsible for the protein modification that happens to the proteins that are being synthesized by the mRNA or the mRNA synthesis and then translation. Again, there are two types. If they have ribosome, they are responsible for protein synthesis translation. But if they don't have ribosome, they are called the smooth endoplasmic reticulum. Then they are mainly responsible for the essential lipid synthesis like phospholipids, cholesterols, hormones, or detoxification in liver or metabolism of carbohydrates. But if you have rough endoplasmic reticulum, then it has got to do with all the protein functionality. For example, protein synthesis, protein folding, protein sorting. Proteins, as I mentioned, they're of different kind, right? Integral, secretory, and different for any other organelle. So all those had been done at the rough endoplasmic reticulum side, while the smooth endoplasmic reticulum is mainly concerned with the lipids. Then you have Golgi bodies. These also we have discussed. So these are these stacked compartments. They have their own cis phase and trans phase. Trans phase will be the one that moves towards the plasma membrane. So they receive the vesicle from this side and transport. On the way, they also modify the protein inside the vesicle, and then they make it ready to be able to be delivered to the destined site. Yeah. So like I was mentioning, so the Golgi body, the functions of these are protein sorting, protein modification in the form of glycosylation, and also proteolysis, in the sense that if they are protein which is being synthesized has not folded properly, then Golgi can send it back to the endoplasmic reticulum so that the protein folding happens properly, or it can decide to just destroy the protein that has been synthesized, and that is why proteolysis. So that's why the forward and the reverse direction of movement, so that the protein is properly folded. Then you have lysosome, which are also what we had discussed as suicidal bags, because they will be responsible for the autophagy. They kill these slice off the components of these cells. If it's in the lumen and it becomes extremely acidic, then you have these different proteases and cytolytic enzymes that degrade the ingested material. And the function of these organelles are to degrade the ingested food material or pathogenic bacteria of any misfolded protein as well. Now, protein targeting is something like where I just mentioned that there are different proteins that need to go to different organelle. So to send them to those different organelle is what is called the protein targeting. And how do you do that? How do you give the signal? One of the way by which you can target a signal, target a protein to a certain destination is by glycosylation. So glycosylation is a form of protein targeting. You add glycosylated groups onto the synthesizing protein, and the protein has the signal where it is to be delivered. So with that glycosylation, it can go to the destined location. And it is important for glycosylation because they are because they are like added to, not the proteins are of different kinds, like they could be enzyme, they could be cell attachment recognition, so they are different functionality that is being carried out by these proteins, and they require these glycoproteins on their surface. So that's how glycosylation is very important. And there are two ways how these glycosyl groups are added to the growing polypeptide chain. They are either O-linked or N-linked. So we'll discuss this glycosylation in detail. So as I've already mentioned, they are generally taken, like they are generally happening in the endoplasmic reticulum of the Golgi apparatus. And what it means is that glycoside means sugar moieties. So the addition of these sugar moieties to the amino acids on the proteins is what is called glycosylation. And generally, the soluble or the membrane-bound proteins there, which are expressed in the endoplasmic reticulum, they go undergo glycosylation so that they become a secreting protein or surface protein or any ligand that is to be go some in the body. And they're also transferred from the Golgi to the cytoplasm. As I mentioned, on the way from the Golgi also they are glycosylated. So as I mentioned that mainly they are O and linked glycosylation, but there are also two other forms that could be C-linked or phosphoglycosylation. So first we'll discuss the N-glycosylation. N-glycosylation and O-glycosylation should be easy for you to guess. It is based on where the glycosyl groups are added. And N and O give away the name of the site of attachment. So N is basically nitrogen. Nitrogen on asparagine residues. So in N-glycosylation, the glycan groups that are added to the nitrogen of the asparagine residue. So on the peptide chain, you would have asparagine. On the asparagine, there would be nitrogen, right? So on the nitrogen, these glycan groups are added. That's why they are N-glycosylation. And as I mentioned, they happens in the ER. When they are being translated simultaneously, this process can happen. The addition of glycan itself takes several events to occur. So the events are first precursor glycan assembly. Now, as you know that ER will have its surface like membrane, and then there is lumen. So there is, first of all, for the glycan to be added to the protein, this has to be first assembled outside the ER. So first step is that there are 14 sugar molecules that come together and get assembled. And that 14s are 3 of glucose, 2 of acetyl glucosamine, and 9 mannose. These 14 sugar, they come together on the ER membrane by dolicol. So dolicol brings like a lipid carrier. So it carries all these different units together and bring it up to the ER membrane. Then the first 7 sugar molecule obtained from sugar nucleotides in the cytoplasm are added. So all these 14 are coming together. First, the 7 are added from the UDPs, and then after this assembly from the ER plasma membrane, these complex is pushed to ER lumen by flipase enzyme. So it will flip, and it will first 7 sugars are added. Then when they are flipped, the other 7 sugars are added, and they will form the glycan here. The glycan is first completed. Now, inside the lumen. So now from after the assembly has happened, 7 outside, 7 inside, then you have this glycan being attached onto the N of the asparagine. So now you see here in the lumen, you have this pre-assembled glycan of different these 14 sugars, right? So now these are attached to the newly formed polypeptide chain by and specific, again a very specific enzyme which with the oligosaccharide transferase. And then the 14-mer precursor glycan is attached to the nitrogen of ASN, and the beta group of serine and threonine residues act as a hydrogen donor for the reaction. So you have the serine and threonine. So like I mentioned, asparagine has to be followed by something and then serine and threonine, because these serine and threonine will give the hydrogen which is responsible for binding of the, or the interaction of these different glycan assembly. So this complex has to be identified, and you already have the glycan. So the glycan can now be added onto this asparagine here, right? So that's why all the N glycans that you see, they will share a common sequence, but they can also be further classified into high mannose and high mannose hybrid or complex glycan, depending on the number of mannose sugar they have added, or what kind of glycan assembly they have taken up. That's what is happening. Okay, now once your glycan attachment has taken place, then you have trimming. So you have these sugar molecules, they are now trimmed by different glycosidases. So you have these glucosidates, right, and mannosidases. So they will chop off some of these residues from the assembly. And this happens both in the Golgi, where you have these mannosides and you have transferases. And it also happens in your ER. And in ER, trimming is done to, so what is the purpose of this trimming? It is done to ensure that the protein is properly folded, and if they have to be degraded. So like I mentioned, that if they're properly folded, they are then transferred to Golgi, and then Golgi again does the same step to check if they are properly folded or not. If folded, then they're transported further to the cytoplasm site, but if they are not, then they are sent back to the endoplasmic reticulum to carry forward this reaction if it can be possible, or they tag it for lysis. The last step is the maturation. So the glycogen maturation. So in the Golgi bodies, after trimming has been done, there is addition of further sugar molecules to generate the diversity on the basis of how it will be high mannose, hybrid, or glycans. Now, when the glycan is accessible to the Golgi mannosidase or transferases, they will start forming these complex oligosaccharide, which are then added to these glycan assembly to give rise to the glycosyl group that is destined for that protein in question. So that's how N-glycosylation will take place. And then after this, from the Golgi, they will be channeled to the destination that they are targeted for. The second type is O-glycosylation. O is because of the oxygen. So here you have the transfer of an acetyl galactosamine on the beta oxygen, or basically hydroxide group of the serine threonine. So again, on the second serine threonine, the hydroxyl group is where these glycosyl group, or here for example this is acetyl galactosamine, is added. So that's how you remember where it is added, which site it is added on. And they are also very important. They are found in mucus secretion proteins and or in the proteins which are part of extracellular matrix and antibody components. And so you add galactosamine, and you can also add, like they're also added with glucose, xylose, and mannose, depending on what type of cell they are or what type of protein they are. And similarly here as well, you can see that there are multiple steps of acquiring the glycan assembly, adding in or and when modifying it to give the final shape. But the only thing is, this is very less complex as compared to the N-glycosylation, where you have mannose. Then the mannose is again they're added, they're removed in very different ways so that they can give rise to a very complex glycosylated structure. Other than that, you have phosphoglycosylation and C-glycosylation or carbon glycosylation. So the first one is phosphoglycosylation, which is generally limited to Leishmania and trypanosoma, where the glycosylation is because of phosphodiester bonds. That's how the name. And the other one is C, glycosylation or only mannosylation. So that is by carbon-carbon binding. It could be only because of mannosiles or mannosyl transferase or mannose only, or it could also involve other complex residues as well. Okay, so On the basis of these topics, there were some questions from the previous year assignments, so I've compiled them and we'll try to solve this. So the first question is, B cells are involved in which of the following type of immunity? Try to remember the like classification of immunity and then see if you can answer this. Second is the bond involved in joining the heavy chain and light chain at the hinge region. Hinge region is where these different polypeptide chains are joined to each other. Which of these bonds are important? The antigen binding site on antibody is which of these? Very important, and try to understand, remember which one is for which one. And the MHC class involved in antigen presentation in macrophages are which? So you try to remember the diagram and see which one was responsible for presenting the antibody uh, in macrophages. So hope we were able to do that. So the B cells are part of humoral immunity, not the acquired immunity, because humoral is where they are mainly because acquired consists both cell mediated and humoral immunity. Second is they are joined by disulfide bonds. Third is antigen binding site on antibody is paratope, but the antigen surface is called the epitope. And the MHC class, which is present for antigen presentation in macrophages, are MHC class 2. Next, the reagent used to screen clone in hybridoma technology for the production of monoclonal antibody is which? Read carefully the name of each of these components. Second is, what would be the blood group if you find no agglutination in for anti-A and anti-B antibodies? Third, the agent causes fusion of spleen and myeloma is which of these? And fourth is the biochemical cell surface marker for detecting apoptotic cells in the flow cytometry through annexin 5 is which of these? Which of these marker? So, like I mentioned, to be careful about the name, the first one was pozanthin aminopterin and not the aminopurine, not the second option, and thymidine. Second is because you see no agglutination for both the antibody virus, that means there is no A antigen or B antigen, so that's why it's blood group O. To fuse the spleen and myeloma cell, we had used polyethylene glycol, so PEG, and it is for the apoptotic cell, so it is phosphatidylserine. Next, which of these following application is not possible with flow cytometry? So it is in isolating green fluorescent proteins or in identifying various cell types, in immunophenotyping or determining the amino acid sequence. Just try to remember the application of flow cytometry. I discussed about these while I was speaking. Next, the channel attributed to granularity of the cell in flow cytometry is which of these? The third is the dye used to analyze cell cycle using flow cytometry is which? Cell cycle cells we are talking about particularly. And this cellular organelle involved in detoxification in liver is which of this? So the first answer is the fourth one is not an application for flow cytometry because you cannot determine the sequence of amino acid. You can know the cell, which cell was present based on the emission pattern, but you can't determine the sequence. Second, for granularity, it was side scatter. For size of the cell, it was forward scatter. Then for cell cycle, we had seen it was propydium iodide. That was the dye that was used. And cellular organelle for detoxification in liver was the smooth endoplasmic reticulum, not the rough. Rough had all the protein sorting and protein trafficking related activities. Okay. Next, the class of pumps used to maintain acidic pH in lysosomes are these calcium plumb or F class, B class, or sodium potassium pump. The next, the organelle adds proteins to glycosaminoglycans to form proteoglycans, and where does it take place? Third, the sorting of newly synthesized protein to their respective cellular destination is which of these? And the last is, where does the ribosome and polypeptide chain complex get targeted once a short segment of polypeptide is synthesized during protein translocation? So where does it actually from, like, once it has this tag, where is it just targeted to? Yeah, the first one is V class pump, which are in the lysosome, which maintain the pH. Second is Golgi complex, as we had seen that they are able to add the glycosaminoglycans to the proteins. And then this first I had mentioned that you target the protein first. So because you're trying to call it the destination location where it is being sent to, it is called protein targeting and not delivery. And then once there is a short segment of polypeptide chain that has been attached onto the growing chain, it is targeted to the endoplasmic reticulum. It is moving out of the nucleus and going to the endoplasmic reticulum. The last set of questions are the binding of signal recognition particle to a large subunit during translocation leads to which of the following process? Translation arrest, transcription arrest, or progress of transcription and progress of translation. Next, protein glycosylation refers to addition of which of these molecules on which of these molecules? Is it of addition of proteins on proteins, or lipids on proteins, carbohydrates or to proteins, or lipids to carbohydrates? Which of these is added during the glycosylation step? Next, the class of lipid transports the dolichol and sugar from cytosolic site to endoplasmic reticulum lumen site. This we had discussed. Try to remember what we had discussed previously. And the next is the transport of protein from endoplasmic reticulum to Golgi complex is mediated by which of these? This also we had discussed the direction and the Cope vesicles which are responsible for the transport. Yeah. So the first is as and when the signal recognition particle is added to the larger subunit, there is arrest of translation because now the post translation modification is will begin to happen. Second, glycosylation of protein means addition of carbohydrates to proteins. Third, flippase enzyme we had discussed, which transferred the assembly of the lipids from the transferred the sugars from the extracellular domain to the lumen of the endoplasmic reticulum. And the fourth is COP2 vesicles are responsible for the movement from endoplasmic reticulum to Golgi. COP1 is for the opposite direction from the Golgi apparatus to the endoplasmic reticulum when proteins are detected for further folding or proper guidance to the protein folding requirement. Okay, so this is where I stopped today, and these topics today we covered about immune systems and the several processes which are involved in the immune system till the final death of the pathogen cells. And then we also looked at the way this transportation happens during in the cell by the different organelles that are responsible. So if you have any questions, you can post it in the comment box, or you can always post it in the discussion forum. Thank you.